Welcome to the Once Changing the World, India's first Future Tech Meets Sustainability podcast. I'm your host, Eddie Havel, and today we have with us Professor Darius Widera, a professor of stem cell biology and regenerative medicine and research division lead at the University of Reading. His lab is mainly interested in adult stem cells and their secretome, inflammatory signaling cascades in health and disease, and novel methods for clinical grade 3D cultivation of human stem cells. His research is focused on the regenerative potential of mesenchymal stromal cells, neural crest-derived stem cells, and pluripotent stem cells in different disorders and conditions. His research aims to tackle disorders and conditions with an inflammatory components such as osteoporosis, lung, brain, and breast cancer. So, Professor, really appreciate you taking time being part of the podcast. I think it'll be great if you could start with a small background, brief introduction, and your works in the space of stem cells and gene therapy.
Thank you very much for your kind introduction, and thank you for having me. So, in fact, my lab is doing a lot of different research projects, focused mainly on adult stem cells. And, my journey focusing on stem cells started actually quite a while ago, whilst I was doing my master's project all the way back in Germany. And I became passionate about stem cells and the potential to basically tackle conditions that we currently cannot really cure using conventional pharmacology treatment options. After completing my master's in Germany, I've moved on and I've done my PhD project in Neurobiochemistry, where I used neural stem cells isolated from the rat brain. And I've investigated how inflammatory signal can actually affect those cells in a dish. From then on, I moved on and I started my postdoc at the University of Bielefeld in Germany, where I work on novel stem cell populations that can be isolated from different human tissues, such as from the nose, from several dental tissues, such as the deciduous teeth. And I look into potential of those stem cells to treat symptoms of Parkinson's disease in animal model of this condition. In 2015, I've relocated my lab to the UK, where I started several projects focused on use of mesenchymal stromal cells in conditions such as osteoporosis, muscle regeneration, but also in collaboration with the University of Science Malaysia in Penang on COPD. This is pretty much my journey in the nutshell.
Could you kind of explain the concept of gene therapy? Because just recently there was this breakthrough which allowed a deaf child to hear properly. So can you maybe just explain the concept of gene therapy and how it's going to impact healthcare?
Of course. So gene therapy allows us nowadays to tackle conditions where we, again, don't have a curative treatment options if we use conventional pharmacology. In order for a condition to be treated using gene therapy, obviously, we need to understand what goes wrong in terms of mutations in the DNA. And in fact, many conditions are caused by mutations. And examples here are cystic fibrosis, even sickle cell anemia, metachromic leukodystrophy, the most recent conditions that has been tackled using gene therapy, and several disorders of the immune system. What basically happens in those conditions, our DNA code is changed, and this leads to several pathophysiological changes in the whole system. Nowadays, since we have discovered the genetic code of the human genome in 2001, we can apply a technique referred to as sequencing to understand what mutations are actually contributing to those conditions. If we know what mutation is causative, then we try to correct this. And gene therapy actually exploits natural DNA repair mechanisms in the cell. As most of you might know, in our cells, we always have two copies of the genes, one inherited from the mother and one inherited from the father. Within our cells, we actually do have DNA repair mechanisms. When mutations are detected by the cell, those natural DNA repair mechanisms kick in, and they use the healthy copy to repair the DNA. Now, these mechanisms can be exploited in gene therapy. We can artificially provide a healthy copy of the gene of interest and exploit the natural mechanisms of DNA repair to basically replace the faulty gene with the correct version. And this is pretty much how all gene therapies nowadays work. In reality, many gene therapies that are used nowadays are in fact combined with stem cells, for example, with blood stem cells. You can isolate the stem cells from the patients, then correct the faulty gene, expand the cells in the dish and transplant it back into the patient. In ideal case scenario, then the gene defect would be corrected and we would cure the patient. And this is pretty much the difference between pharmacological treatment of these conditions where we actually don't tackle the cause, we are only treating the symptoms.
Right. The DNA is basically the source code of life. And, through these tools such as CRISPR CAS9 gene editing tools, you can specifically kind of take out a specific gene or add a gene to it, maybe the gene which is mutating. How is this going to impact the healthcare? Because so far, I think the way we have treated healthcare is bombarding it with, possibly medicines or things like that. But now you're saying that, there's a way to kind of go right down to the root cause, figure out which gene is kind of mutating, which gene is possibly the cause of that specific kind of disease. So how does this impact the future of the healthcare? Because most of the diseases are the underlying nature of is basically a genetic mutation. So how is this going to impact the future of healthcare?
I think gene therapy will have tremendous impact on the healthcare system. In fact, gene therapy for metachromatic leukodystrophy can be already prescribed in the UK on the NHS. So basically the healthcare system is already paying for it. For this condition, there is no curative treatment using conventional pharmacology. Therefore, this investment has long-lasting effects. And in fact, this disease is so severe that the patients have very, very short life expectancy if only conventional pharmacology is used. Therefore, even though gene therapy is quite expensive at the moment, the investment is certainly justified because the patients are cured in ideal case. So this is going to have tremendous impact. What we need to keep in mind is that at the moment, in fact, gene therapy is quite expensive. However, we need to keep in mind that there is economy of scale. If more gene therapies are going to be successful, then the costs will ultimately go down. So I think this is going to have a huge and positive impact on healthcare, as we are going to be able to help patients where we didn't have curative treatment. Another example where gene therapy is already making impact is actually AIDS. Since you mentioned this before, and I didn't cover this in detail, gene therapy can actually replace faulty gene, but we can do the opposite. If actually a healthy version of the gene is contributing to the disease, we can actually actively remove it. And this is what is currently being done if we think about gene therapy for AIDS and infection with HIV. HIV uses a very specific receptor in the cell surface of our immune cells to enter the cell. Now, it has been actually discovered a while ago that certain percentage of Caucasians are immune from infection with the HIV. And the cause is because those Caucasians carry a certain mutation in the receptor in immune cells, and the receptor of the interest here is CCR5. And the idea of the gene therapy was basically to actively mutate this receptor to prevent HIV from entering the cells. And in many clinical cases, this therapy has been successful. So now, in conventional pharmacological treatment of AIDS, patients have to take quite a lot of drug, in simple terms, for the lifetime. They do have quite a long life expectancy, but this actually produces huge costs to the healthcare system. If we can replace this lifelong application of expensive drugs by one-off, possibly quite expensive gene therapy, then obviously this would offset those costs of lifelong prescription of drugs. So I think the balance is going to be there nevertheless.
You mentioned about being very, very expensive. How and when do you think this is going to be democratized, where it's accessible for everyone? And in the genetic therapy treatment space, there is already sickle cell treatment. There is deaf children have been, able to hear, and there is various other spaces. I mean, how do you get this, to be accessible for everyone? What would it take?
First of all, future is very hard to predict, but let me start with a slightly different example. Back in 2001, when we started to sequence the human genome, the costs for a single sequencing were roughly $100 million. Approximately 20 years later, the costs went down to $1,000 per genome. So again, technology is evolving. We are getting more and more efficient. Technology is getting cheaper. So if I would have to make a prediction, I think the situation will look very, very different in 20 years' time, once those technologies are more established. And in fact, new approaches to gene editing are being implemented on a daily basis. As you mentioned before, the most common approach to gene editing nowadays is CRISPR Cas9. But let's keep in mind that this is not the first generation of gene editing. We started quite a while ago with technologies such as zinc finger nucleases, followed by tail ends, and all of those technologies have been used in the clinics. However, they were quite expensive. Nowadays, CRISPR-Cas9 allows us to edit multiple genes at the same time, thereby reducing costs already now. And I certainly believe that the technology will move forward, will become more efficient, but we need to. So if I would have to make a prediction, I think it's going to be democratized in roughly 2 decades, possibly earlier.
Really, really wish for that, to happen, like maybe possibly sooner. Can you explain the concept of regenerative medicine? Do share some of your works in this field of regenerative medicine.
Yes, regenerative medicine is an umbrella term for pretty much many different curative treatments. What is considered regenerative medicine are things like bone marrow transplantation. Then you could boost the endogenous regeneration in the patient. This would be regenerative medicine. In fact, gene therapy also falls under the umbrella of regenerative therapy. Then think things like organ transplantation, even xenotransplantation, where we transplant animal-derived organs into the humans. This is regenerative medicine as well. However, nowadays, regenerative medicine mainly involves combination of some of those aspects. In fact, quite interesting and I think groundbreaking application of regenerative medicine was development of a curative treatment for epidermolysis bullosa quite recently. Epidermolysis bullosa is a degenerative condition of the skin where the carriers of certain mutations in collagen or laminin have chronically inflamed skin. They tend to develop blisters and are basically in chronic pain. Until very recently, we did not have any curative option for these disorders. However, I think it's been 2 years ago, a new therapy has been applied in a patient where the scientists and physicians isolated skin cell from the patient, expanded them, corrected the respective mutation, then differentiated those stem cells into different types of the cell, applied 3D bioprinting to create artificial layers of skin, and then transplanted the skin into the patient. And believe me, the outcomes were phenomenal. So we are already there, and regenerative medicine is becoming a reality. And this was really, really exciting.
Right. So I mean, there have been animals, which have shown like great regeneration potential, right from your lizards to axolotls and many other these animals. We humans also have some kind of a regeneration capabilities, though that it has vanished over the ages, I mean, so how do you see this kind of panning out? I mean, what's been the learnings from the animals that's been taken to kind of leverage it for these regenerative medicine purposes?
This is a very, very good question, and well, fits quite well with your background. Sounds a bit like sci-fi. If we basically think about boosting endogenous regeneration of the humans, human tissue, we need to be quite careful, since animals like axolotl, in fact, can regenerate a lot, but they don't develop diseases such as cancer. And this is quite interesting. In fact, many of natural mechanisms linked to regeneration are also upregulated in cancer. So it's going to be very, very hard to find a balance. And I would be very, very careful in developing similar therapies. In fact, any transplantation of stem cells, they can differentiate into many different cell types, is already associated with the risk of development of tumors. And this is actually more the case if we think about pluripotent stem cells compared to adult stem cells. So I think in the human system, this is quite tricky. I think it could be done, but the potential side effects can be quite severe. But this is my personal opinion, and obviously this could be feasible, but we need to consider the risks and dangers of this approach as well.
Right. Can you talk about mesenchymal stromal stem cells and their regenerative potentials and possibly their potential applications for therapy?
Mesenchymal stromal cells, how I prefer to call them, have been discovered quite a while ago. And the first characterization of those cells was actually using morphology. The researchers originally discovering the cells recognized they resemble fibroblasts quite a lot. Then there was a lot of research done on those cells, and it has been shown that at least in a dish, those cells can form bone cells, cartilage cells, fat cells, and therefore the idea evolved that those cells are actually stem cells. This was followed by quite huge hype in the scientific community, and plenty of papers claimed that those cells can give rise into cell types such as neurons. However, not a single functional proof has been published basically supporting this claim. If we have such promising data on the threads from the lab, obviously this encourages a lot of clinical evaluation of the technology, and in fact, quite a lot of clinical trials showed a very good safety profile for MSCs and also surprisingly good clinical outcomes. However, after more careful evaluation, it has been discovered that those cells, if transplanted, do not survive in the patient for very long. In fact, if injected into the patient, most of the cells are being trapped in the lung or in the liver. And the conclusion here was that those cells do help in certain conditions. However, the mode of action is not that those cells replace the tissue that is degenerating, but they support endogenous regeneration. And those cells so by secreting certain factors that improve regeneration of the patient. A few years later, it has been discovered that those paracrine effects, how we call them, are caused by tiny vesicles secreted by those cells referred to as extracellular vesicles. Now, quite few follow-up research projects have shown that extracellular vesicles secreted by MSCs have very, very similar clinical outcomes compared to transplantation of MSCs. So nowadays, we actually do not believe that those cells are stem cells. And even Kaplan, who was the scientist who initially coined the term mesenchymal stem cells, suggests to refer to those cells as medicinal signaling cells. So the abbreviation is going to be still the same, but the meaning, after we know the mode of action, is very, very different. So I personally don't believe those cells are stem cells, but they certainly do have clinical use.
Right. Can you explain what neural crest-derived stem cells are and how you're leveraging it in treating neurodegenerative diseases?
Of course, in order to understand potential of neural crest-derived stem cell, we need to go back to developmental biology. Once an egg cell is fertilized by a sperm cell, this egg cell starts obviously to divide. After several rounds of division, we see a process referred to as formation of germ layers. In all mammals, we have three distinct germ layers, namely ectoderm, mesoderm, and endoderm. And different cell types originate from each of those germ layers. During development, our cells specialize more and more, and as a result, they lose differentiation potential. For example, pluripotent cells, such as embryonic stem cells and induced pluripotent stem cells, can differentiate into cells of all three of those germ layers. So in theory, they can replace all of cells in our body. In contrast, adult stem cells can only differentiate into the germ layer of origin. For example, if you isolate a stem cell from the brain of an adult, this stem cell is only going to be able to differentiate into neurons or astrocytes, but cannot differentiate into, let's say, blood cells. Vice versa, if you isolate a blood stem cell, this stem cell is going to be able to give rise to most of the cell types within the blood system, but is not going to be able to replace neurons. Good. This is pretty much the picture during development and potential of stem cells. Now, neural crest-derived stem cells are quite unique. Those cells appear actually quite late during the development. They migrate out of the niche and differentiate into cells of at least two germ layers. They can differentiate into cell types such as peripheral neurons, but also into mesodermal cells such as muscle, bone, and fat cells. Interestingly, some of those adult neural crest cells persist until the adulthood, especially in the head region, in the craniofacial region, which originates nearly exclusively from neural crest. We can isolate those cells from tissues such as our nasal tissue, from our oral cavity, and if we isolate them and expand them, they still have the potential to differentiate not only into bone, fat, and cartilage tissue, but they can also replace peripheral neurons, and this makes those cells quite unique. There were quite few attempts to use neural crest-derived stem cells to treat conditions, for example, like spinal cord injury. So in spinal cord injury, the connection is actually interrupted, and those cells have been used to basically bridge the gap and reestablish the connectivity with, I would say, mixed results. What's quite interesting, though, is that although those cells can in fact differentiate into neurons, the main mode of action is quite similar to the mode of action of mesenchymal stromal cells that we've discussed before. Those cells also help to boost endogenous regeneration. So they have both. They have the ability to differentiate and replace lost tissue, but also to improve the regeneration via secretion of certain protective factors. So those cells can be, in fact, used in many conditions.
What are the challenges in targeting cancer stem cells, and how can gene therapy be utilized to overcome these challenges?
This is a very, very interesting question and very interesting concept. In order to understand cancer stem cells, we need to think a bit about conventional therapies that are currently used against cancer. So if it comes to cancer, the most common therapy option is obviously surgery. However, if you use surgery, there is always the risk that we miss some cancer tissue, and this could lead to relapse. Therefore, in many cases, surgery is combined with chemotherapy and radiation. Now, chemotherapy and radiation are therapies that usually target only dividing cells. And this also explains many of side effects of those therapies, like hair loss or anemia, where chemotherapy is not only targeting dividing cancer cells, but also dividing blood stem cells and so on. In order to understand cancer stem cells, we also need to think about the role of stem cells in the healthy body. In the healthy body, stem cells are actually not very busy. They remain dormant. They do not divide. Unfortunately, in cancers, which are intrinsically heterogeneous, there are also cells that resemble stem cells. They remain dormant and they do not divide. Therefore, if we apply chemotherapy or radiation, we will only hit dividing cancer stem cells, whilst cancer stem cells remain hidden. After a while, those cells, however, can re-enter the cell cycle, start dividing, and this could lead to relapse. And unfortunately, this is something we see in the clinical environment quite frequent. The idea of therapy that would specifically target cancer stem cells can be to deliver cancer therapeutics specifically to cancer stem cells. And this can be exploited by utilizing what we already know about cancer stem cells. Cancer stem cells differ compared to both healthy cells and also differentiated cancer cells. They do express unique surface receptors, and this can be used to develop new therapies. You can, for example, engineer an antibody which carries chemotherapeutic drug, and this antibody could then bind to cancer stem cell specific receptors and deliver chemotherapy specifically to cancer stem cells whilst leaving healthy cells unaffected. I personally do not believe that gene therapy is the most promising way to go, since what we also need to keep in mind that cancer usually does not originate because a single mutation in a cell occur. In fact, there's quite famous hypothesis about cancer development formulated in the 70s by Knudsen. It's still called the Knudsen hypothesis. And the Knudsen hypothesis claims that it takes at least 2 mutations, and in many cases even more, to lead to cancer. And using his own words, it takes at least 2 to tango. Therefore, we would need to know exactly what mutations those cancer stem cells are actually carrying, and this could be then used to tackle them. In fact, they are quite modern approaches to treat cancer based on vaccine strategies developed during the COVID pandemic. I'm referring specifically to mRNA-based vaccines. In fact, the same companies who use this technology to tackle COVID, namely Moderna in the United States and BioNTech in Germany, are currently running a lot of clinical trials focused on personalized treatment of cancer. What those companies are doing, they are isolating cancer tissue and healthy tissue, then sequence the genome of both cancer cells and healthy cells, identify what mutations are actually those cells are actually carrying, and design personalized cancer vaccines for each patient. And I think this is the way forward. However, at this very stage, we don't have the technological means to isolate cancer stem cells, heterogeneous cancers, to treat them individually. So at the moment, we do have personalized treatment options, but they would tackle all of the cancer cells, and this includes cancer stem cells that were previously hidden, because they will carry very similar mutations to differentiated cancer cells. So this is not specifically a gene therapy, but personalized cancer vaccination, which is based on genomics.
There are a lot of these groups, researchers and scientists who are also working on organoids, where they're creating organoids of liver, brain, and various other organs to kind of understand it better, to build better therapies. Would you like to share your insights on that, organoids?
Yes, I personally believe that organoids are actually the next big step forward. So if we think about conventional 3D cell culture, we usually only use very few different cell types. Again, organoids resemble the physiological situation way closer than conventional 3D cell cultures, where you have only a very limited number of different cell types. However, our organs are composed of many different cell types that all basically talk to each other. They all influence each other. And the organoid systems are basically mini organs, or well, structures resemble mini organs where we can study in many things in more physiological environment. And so far, many different organoid systems have been developed, starting from, as you mentioned, brain organoids. We do have gut organoids, lung organoids, and this allows us to do quite few things that we couldn't do before. We can now study early development. We can develop drugs in more complex models. And in fact, organoid technology is adapted nowadays quite wide by the pharmaceutical industry as well. In addition to this, organic technology has been also revolutionizing what we already know about embryonic development of the human body as well. Since quite recently, and it was quite big in the media in 2023, several independent groups around the globe have developed models of the early embryo. And this pretty much is based on the organoid technology, where we can actually understand early birth defects way better than we used to do in the past.
Right. Do you see that in the future, maybe through these organoid technologies, maybe you could possibly build large-scale organs?
At the moment, the limitation is obviously the size. What you need to keep in mind that if the organoids reach certain size, they will basically starve. So what we need to do, we need to supply them with nutrients. How does our body deal with this problem? We have blood vessels. So in order to develop organs, well, as you suggested, as potential replacements for organs and for transplantation, we would need to make those organs bigger and way more complex. And this would certainly rely on angiogenesis. We would need the formation of blood vessels. We are not there yet, but I personally believe that this is possible. But there are also other considerations. Obviously, self-organized systems resemble the real thing, but they are not identical. Also, and this is independent of the type of the organoid, the efficiency of formation of organoids is still very low. So we need a lot of improvement. We need optimization of the methods. And obviously, at some point, we will face the problem of the size. So we need to make the models even more complex by implementing perfusion, implementing blood vessels. It is doable, but we are not talking about development in the next couple of years. This will certainly take time.
Right. Can you elaborate on the therapeutic potential of extracellular vesicles and the applications in regenerative medicines?
This is actually quite a modern approach to regenerative medicine, since the use of extracellular vesicles is sort of based what we have learned from stem cell transplantations. Many of stem cell transplantations in the past have shown beneficial clinical effects without any signs of integration and differentiation. So this led to the idea that those cells must be secreting something beneficial, and this can be exploited. In fact, many clinical trials that have been conducted using extracellular vesicles show similar beneficial effects compared to stem cell transplantation. So nowadays, many labs, including my lab, are working on protocols and methods where we could substitute stem cell transplantation by transplantation of extracellular vesicles. And this makes sense if the clinical outcomes are very similar. Using extracellular vesicles instead of stem cells could have several advantages. And first advantage would be obviously standardization. We are all different, and as a result, our stem cells are also very different. Therefore, the efficacy of stem cell therapy is going to be very different from batch to batch. If we now utilize extracellular vesicles, this could lead to more consistent therapies if we, for example, would use extracellular vesicles isolated from stem cells from multiple donors. We could also reduce the risk of side effects. Obviously, if we don't transplant stem cells, then the tumor formation risk is going to be massively reduced compared to transplantation of living tissue. And this would also reduce the cost of the therapy. So there are certain advantages. However, limitations now are upscaling. We are not there yet, and production of extracellular vesicles tends to be quite cost and labor intense. So this is certainly something we need to address in future. Also, regulatory aspects are not very clear. How should the regulators actually treat this therapy? Is it similar to stem cell therapy? Is it similar to antibody-based therapy? No one really knows at this stage, since this field is actually so new. So there is certainly action needed, not just from the scientists to improve the therapies and the protocols, but also from regulators to ensure patient safety.
As a scientist, as a researcher, as somebody who's leading a lab, what do you think are the challenges or the limitations of taking your research from lab to the market?
If it comes to translation of basic research into the clinic, we are facing quite few hurdles. And again, this starts from translation, what we have learned in animal models, to the human system. Obviously, mice are not humans. So there are always risk of basically side effects that we didn't see in the animal models if we translate it into the clinical trial. So this is the first risk, obviously. Then there are also cost implications. Clinical trials are very expensive, and pharmaceutical industry is notoriously risk averse. So the money needs to come from somewhere else. Also, if you translate quite new groundbreaking therapy, you have to deal with quite a few regulatory issues, whilst you need also to ensure that the patients are going to be safe. And this is something extremely important. What I have noticed in the last 20 years is quite a dangerous trend. All of the hypes that we've discussed before have been, in fact, exploited by shady businesses around the globe. If the regulators don't act quickly enough, this ultimately leads to exploitation of all of the hypes by direct-to-consumer business. And we've seen this if it comes to mesenchymal stem cells around the globe. And unfortunately, the same is already happening with extracellular vesicles. Shady businesses are offering those unproven and unregulated therapies directly to the patients, even before we have clinical evidence for safety and efficacy. This actually produces quite a lot of risks, and there were reports on many, many patients actually harmed and even killed by similar businesses around the globe. Quite recently, a patient in the United States has actually died after receiving stem cell therapy in Mexico. So again, those businesses putting not only the patients at risk, but also the whole scientific community and all of the honest researchers who are working in this field. Since, well, if something really, really bad happens, this would basically put the whole scientific area and all researchers working on therapies in very, very bad light. So this could lead to credibility crisis. So I think there is certainly action needed from the regulatory side all around the globe.
Right. Would you like to share any of your updates from the lab or any research that you're currently doing?
Obviously, I can't comment on any details on unpublished work, but just to give you an idea, what we are currently doing, we are combining all of the aspects that we discussed before. So what we want to do, we want to make extracellular vesicles secreted by stem cells more efficient. And we are doing so by using the, in my opinion, most promising adult stem cell type, neural crest-derived stem cells. We have made them immortal to produce extracellular vesicles continuously. We do this in 3D, and we combine this 3D cell culture with electrical stimulation to make them even more potent. And just to give you a short spoiler without too many details, we are using those extracellular vesicles to tackle conditions such as lung cancer, brain cancer, and breast cancer. And the initial data sets are very, very encouraging.
We wish you and the team the very best, Professor. Really, really appreciate you taking time and being part of the podcast. My last question to you: how do you envision the future of stem cells and gene therapy, and what advancements do you anticipate in the field in the next coming years?
I certainly expect that more combination therapies, such as the combination that we discussed before for epidermolysis bullosa, will become reality. I can imagine that more and more regenerative medicine therapies will combine differentiation of stem cells into complex and different cell types, combined with bioprinting, bioengineering. So we will combine stem cell technology, gene therapy, bioengineering to have more and more complex approaches to treat more and more complex conditions. I also expect that in near future, we are going to be able to tackle conditions that are caused not just by one mutation, but by several, since we are developing more technologies allowing us to perform multi-site gene editing. And if we combine this with recent developments in organoid technology, stem cell technology, 3D cell culture, bioprinting, this will allow us to tackle conditions and disorders that we could not treat before. In fact, if you observe and watch the media carefully, then you will see that news on new therapies are popping more and more frequently month by month. And this is actually very, very encouraging. And what's also encouraging is that regulators start to approve those therapies, and that also healthcare system are already willing to pay for this type of revolutionary and groundbreaking technology.
Professor, really, really appreciate you taking time and being part of the podcast. Yes, I think we're sitting in a fantastic point of time, right from genetic sequencing. I think we'll get into a world where every individual's genomes is going to be sequenced, which will lead to personalized medicine. AI and machine learning is going to play such a huge role with AR, VR, MR, where augmented reality, virtual reality, rather than us going to the healthcare will come to us. So I think we're sitting in a fantastic point, and more power to you and people such as yourself. And I really hope that we go towards building healthcare, which is really revolutionary and which really takes the world forward and makes us disease-free. So really appreciate you taking time and being part of the podcast. For my listeners, if you like what you see in here, then please press the subscribe button. Until next time, see you guys. Bye-bye. Thank you.
Thank you for having me.